Hello everyone and welcome back to our channel. Today, we're diving deep into the microscopic world to explore one of the most vital components of life, the cell membrane. It's the gatekeeper, the security guard, and the communication hub for every single cell in our bodies. So, let's get started. First off, what exactly is the cell membrane? Also known as the plasma membrane, it's the incredibly thin outer boundary of a cell. Think of it as the cell's skin. It separates the internal world of the cell from the outside environment. This separation is crucial because it allows the cell to maintain its own unique, stable internal environment, protecting its integrity and making sure it can function as an individual unit. Without this membrane, a cell wouldn't really be a cell at all. It would just be a jumble of molecules mixed with its surroundings. Now, you might be wondering, what does this amazing structure actually look like? Well, that's a bit tricky. The cell membrane is so incredibly thin that you can't see it with a standard light microscope. It's simply beyond the resolving power of light. To even get a glimpse, scientists need to use special staining techniques that highlight it. To truly appreciate its structure, we need to turn to a much more powerful tool, the electron microscope. Under an electron microscope, we can see that the cell membrane is only about 7.5 to 10 nanometers thick. That's astonishingly thin. When prepared for viewing with a chemical called osmium tetroxide, the membrane reveals a beautiful and distinct pattern. It shows a trilaminar appearance, which means it has 3 layers. There are 2 dark, electron-dense outer layers with a lighter, electron-lucent layer sandwiched in between. This 3-layered look gave scientists the first major clue about its molecular organization. So, what is the cell membrane actually made of? Its fundamental structure is described by the fluid mosaic model. The membrane is primarily composed of a lipid bilayer, which acts as a flexible fluid foundation. Embedded within or attached to this lipid sheet are various proteins, and on the outer surface, you'll find carbohydrates. It's like a mosaic, with different pieces coming together to form a functional whole. Let's break down these components, starting with the lipids. There are 3 main types of lipids in the cell membrane: phospholipids, glycolipids, and cholesterol. The most abundant are the phospholipids. Each phospholipid molecule has a very specific design. It has a head that is polar and hydrophilic, meaning it loves water, and 2 tails that are nonpolar and hydrophobic, meaning they fear water. Because the inside and outside of a cell are watery environments, these phospholipids arrange themselves in a clever way. They form a bilayer or a double layer. The water-fearing hydrophobic tails all turn inward, facing each other and creating a nonpolar core for the membrane. Meanwhile, the water-loving hydrophilic heads face outward, interacting with the aqueous fluid both inside the cell, the cytoplasm, and outside the cell. This spontaneous arrangement is what forms the basic fabric of the membrane. And that trilaminar appearance we saw under the electron microscope. That's because the osmium stain binds to the hydrophilic heads, making those 2 outer layers appear dark. Next up, the proteins. If lipids form the structure, proteins are the real workhorses of the cell membrane. They are the major functional components and carry out most of the membrane's specific tasks. We can classify membrane proteins into 2 main groups: integral proteins and peripheral proteins. Integral proteins, also called transmembrane proteins, are firmly embedded within the lipid bilayer. Most of them are so large that they span the entire width of the membrane, with parts exposed on both the inside and the outside of the cell. These proteins have a huge range of functions. Some act as pumps, using energy to actively transport ions and molecules across the membrane. A classic example is the sodium-potassium pump. Others form channels, creating pores that allow for the passive passage of specific ions, water, and other small molecules. Integral proteins also function as receptors. They have specific shapes that allow them to bind to signaling molecules, or ligands, on the outside of the cell. This binding event triggers a response inside the cell, which is fundamental for cell communication. Some are linker proteins anchoring the cell's internal skeleton, the cytoskeleton, to the external network of molecules called the extracellular matrix. Others are enzymes like ATPase, which helps power the ion pumps. Finally, some are structural proteins that help hold cells together, especially at cell junctions. The second group is peripheral proteins. Unlike their integral cousins, these proteins are not embedded in the lipid core. Instead, they are more loosely attached to the surface of the membrane, usually on the inner, cytoplasmic side, but sometimes on the outer surface as well. They often bind to integral proteins or to the polar heads of the phospholipids and play supporting roles in cell signaling or anchoring the cytoskeleton. Last but not least, we have the carbohydrates. These are found exclusively on the external surface of the cell membrane, creating a sugary coating called the glycocalyx or cell coat. These carbohydrates aren't just floating around. They are attached to either proteins, forming glycoproteins, or to lipids, forming glycolipids. The glycocalyx is incredibly important. It acts as a sort of molecular ID badge for the cell, allowing the immune system to recognize self-cells and distinguish them from foreign invaders like bacteria. It also protects the cell from mechanical damage and helps cells stick to each other. So, with all these components working together, what are the overall functions of the cell membrane? As we've touched on, it's all about control and communication. It maintains a constant and optimal internal environment for the cell's machinery to work. It helps preserve the cell's shape. Critically, it acts as a selective permeability barrier, meaning it controls what enters and leaves the cell. It's like a bouncer at a club, deciding who gets in and who stays out. It also enables the cell to recognize specific molecules, like antigens, and to receive signals from its environment, allowing it to respond and adapt. These amazing functions, especially transport, recognition, and signaling, are achieved through three main mechanisms: membrane transport, vesicular transport, and signal reception. Today, let's focus on the first one, membrane transport. This is the process of moving substances across the cell membrane. There are two major types of membrane transport: passive transport and active transport. Passive transport is the easiest way for substances to cross the membrane. It is called passive because the cell doesn't need to expend any energy to make it happen. Movement occurs naturally down a concentration gradient, which means substances move from an area of higher concentration to an area of lower concentration, just like a ball rolling downhill. There are a few forms of passive transport. The simplest is osmosis, which is the specific movement of water across a selectively permeable membrane. Then there's simple diffusion. This is for small, uncharged molecules that can slip directly through the lipid bilayer. Gases like oxygen and carbon dioxide, as well as lipid-soluble molecules, use this route. Some small molecules, like water and certain ions, can also pass through protein channels via simple diffusion. Finally, we have facilitated diffusion. This is still passive, so no energy is used, but it requires the help of a specific protein. It asks for molecules that are too large or too charged to diffuse through the lipid bilayer on their own, like glucose and amino acids. These molecules bind to a carrier protein, which then changes shape and shuttles the molecule across the membrane. It's like having a special escort to get you through the gate. The second major type of transport is active transport. As the name suggests, this process requires energy, usually in the form of ATP. Why? Because active transport moves substances against their concentration gradient from an area of low concentration to an area of high concentration. It's like pushing that ball back up the hill. This process always involves protein pumps that use energy to change shape and force molecules across the membrane. The most famous example, which we mentioned earlier, is the sodium-potassium pump, which is vital for nerve function and maintaining cellular balance. And that's our detailed look at the cell membrane. From its basic definition to its intricate molecular structure and its crucial roles in transport, the cell membrane is a dynamic and essential part of every living cell. It's a perfect example of how structure dictates function in biology. Thanks so much for watching. If you found this video helpful, please give it a thumbs up, and don't forget to subscribe to our channel for more deep dives into the fascinating world of science. Hit that notification bell so you won't miss our next video. See you next time.